Hello everyone, in this video we will discuss transport of substances across the plasma membrane from the region of its higher concentration to lower concentration and from the lower concentration to higher concentration with or without expenditure of energy. Based on this, transport is of majorly two types, that is passive transport and active transport. Sir, what is passive transport and active transport? If substances passes across the plasma membrane without the expenditure of energy, then it is called as passive transport. If substances pass across the plasma membrane without the expenditure of energy, then it is called passive transport. If substances pass across the plasma membrane with the expenditure of energy, then it is called active transport. If the substances pass across the plasma membrane with the expenditure of energy, then it is called active transport. Now, first let us talk about passive transport. Here, passive transport is of mainly two types, that is diffusion and osmosis. Now, first let us talk about diffusion. Transport of substances and these substances can be in different states like solids, liquids, gases. Transport of substances, that is solids, liquids, gases, from the region of its higher concentration to the region of its lower concentration is called as diffusion. And generally the diffusion is faster in gases, slow in solids. Diffusion is faster in gases and slow in solids. And we can understand medium in liquids. And this diffusion process occurs in both living and non-living things. It occurs in both living and non-living things. So now let us take one example. How is this diffusion take place in the living cells? Diffusion across the cell membrane. So now let us take one example, a unicellular organism, amoeba. Here we know that amoeba is the unicellular eukaryotic organism and it live in fresh waters. We know that in water gases are present in dissolved state. So in aquatic region the oxygen is present in dissolved state, and this amoeba require oxygen to perform respiration. So the oxygen present in water move from its higher concentration to lower concentration by the simple diffusion process. And at the same time, during cellular respiration, whatever the carbon dioxide is produced in cellular respiration, its concentration inside the cell cytoplasm is higher. So this carbon dioxide passes across this plasma membrane of this amoeba from its higher concentration to lower concentration. So this is how the metabolic gases like carbon dioxide and oxygen move out and inside the cells by the simple diffusion process. Now let me write here, in amoeba metabolic gases, that is CO2 and oxygen, move out and into the cells by diffusion. So here we can see that exchange of gases done by the diffusion across the plasma membrane. Exchange of gases is done by diffusion across the plasma membrane. Similarly, if you see in multicellular organisms like in humans, exchange of gases takes place between the membrane of lung alveoli and blood capillaries by the simple diffusion process. So this is how the exchange of gases done by diffusion process across the plasma membrane in all the living cells. Now let us see the significance of diffusion. Exchange of gases that is O2 and CO2 during respiration and photosynthesis occurs through diffusion process. This is the significance of diffusion. Here significance means importance of diffusion process. In photosynthesis and respiration process, the exchange of gases takes place by the diffusion process. And if you see one more example, aroma of flowers spread due to diffusion of volatile aromatic compounds in air. Here you can take example of such plants like jasmine. Wherever a jasmine plant is present and flowers in that jasmine releases a good smell that will spread from its higher concentration to the lower concentration. So, this is the significance of diffusion in living organisms. And this diffusion process also occurs in non-living things. For example, if you open a scent bottle in the closed room, after some time the smell spread throughout the closed room. So, this is because of the substances are moving from the region of their higher concentration to lower concentration by the diffusion process. Now, let us discuss about another type of passive transport, that is osmosis. Osmosis is a type of diffusion. Here what happens is the diffusion of water or solvent across semi permeable membrane from the region of its higher concentration to the region of its lower concentration is called osmosis. So, this osmosis process occurs only in the solvents, this we should remember. And here the movement of solvent, that is water, from the region of its higher concentration to the region of its lower concentration is called as osmosis. Now, let us take examples of osmosis. Generally, we know that the plant roots absorbs water, and this absorption of water by plant roots occur by the osmosis. And the another example is absorption of water by unicellular freshwater organisms like amoeba, they live in water. Absorption of water by unicellular freshwater organisms. Now, let us see under this types of osmosis: endosmosis, exosmosis. So, here what is meant by endosmosis and exosmosis? Endosmosis is the osmotic entry of water into a cell or a system. It is the osmotic entry of water into a cell or a system. is called as endoosmosis. Here endo means entry of water inside the cell, and here exosmosis, exo which means exit or withdrawal. So exosmosis is the osmotic withdrawal of water from a cell or a system is called as exosmosis. Plant and animal cells placed in salt or sugar solution will behave in different ways depending upon the concentration of external solution. Plant and animal cells placed in salt or sugar solution will behave in one of the following ways depending upon the concentration of external solution. So based on this, solutions are of different types. Now let us talk about types of solutions. So first, what is meant by solution? Here it is. Solution is the combination of solute and solvent. For example, if you take a sugar solution or a salt solution, here I am taking sugar solution. In this, sugar is the solute and water is the solvent. Now here let us talk about types of solutions. The first one is hypotonic solution, isotonic solution, hypertonic solution. Hypotonic solution. If the external solution has more solvent and less solute than the cell, then the external solution is said to be more dilute than the cytoplasm of the cell, then it is called as hypotonic solution. Here the name hypo, which means solvent is more and solute is less. If the external solution has more solvent and less solute, than the cell, then the external solution is said to be more dilute than the cytoplasm of the cell. Now let us see isotonic solution. If the external solution has similar solute or solvent concentration as the cell, then the solution is said to be isotonic solution. And the last one is hypertonic solution. If the external solution has more solute and less solvent than the cell, then the solution is said to be more concentration than the cytoplasm of the cell. So this is about types of solutions. Now here let us see movement of water in a cell. Movement of water in a cell in different types of solution. In different types of solution. Here I have taken both animal cell and plant cell, and here are the three type of solutions. And we know that inside the both plant and animal cells, the cytoplasm contain different type of components. And when you see these three different types of solutions, in hypotonic solution the solute is less and solvent is more. In isotonic solution both solute and solvent concentration is similar as the cell cytoplasm. In hypertonic solution solute is more and solvent is less. Now let us see what happens when we place animal cell and plant cells in these three different types of solutions. How is this movement of water takes place in these three different type of solutions. Now let us start with the animal cell placed in hypotonic solution. When we place animal cell in hypotonic solution, the solvent is more inside the external solution. So when we place an animal cell, that is red blood cell, see here it is the RBC. Is membrane present in RBC? Yes. The membrane of RBC is called as Donan's membrane. It was discovered by a scientist, Donan. So it is called as Donan's membrane. And when we place RBC in hypotonic solution, first there is a movement of water from external solution to inside the cell occurs, and this movement of water inside the cell is called as endosmosis. Osmotic entry of water inside the cell is called as endosmosis. So after some time, this continuous entry of water inside the cell causes pressure on the plasma membrane so that it cannot tolerate increased pressure of water on this membrane, so that it results in bursting of the membrane of RBC. After some time, we can see there is a bursting of RBC, and we call this process of bursting of RBC when it is placed in hypotonic solution as hemolysis. This is the red blood cell. When it is placed in hypotonic solution after some time, due to the entry of water inside the cell, the pressure caused on the membrane of the RBC, and it results in bursting of RBC, and this process is called as hemolysis. And if it is any other cell instead of RBC, then we can use this term lysis. So here RBC is hemolyzed, and when the same RBC placed in hypotonic solution, here the osmotic entry of water inside the cell is equals to the exit of water outside the cell. So in such cases, there is no change in the cell shape or size. So the cell would be in normal. Now let us see, when we place RBC in hypertonic solution, we know that in hypertonic solution, the external solution has more solute and less solvent than the cell. That means inside the cell amount of water is more than the external solution. So in such case, the osmotic exit of water takes place. So we call this process as exosmosis. And it results in the membrane of RBC shrinks or shriveled. This shrinking of RBC is taking place due to the exit of water from the cell into the external solution, because the external solution has more solute than the cell. So it results in shrinking of the RBC. This process is called as crenation. The shrinking of RBC when it is placed in hypertonic solution, this process is called as crenation. Now see here, when we place RBC in hypotonic solution, it undergoes the hemolysis, that is bursting of RBC. When the RBC placed in hypertonic solution, it shrinks or shriveled. This process is called as crenation. And when the RBC placed in isotonic solution, no change in it. Now let us see what happens when plant cell is placed in these three different types of solutions. When a plant cell is placed in hypotonic solution, we know that in hypotonic solution solvent is more. So here there is a osmotic entry of water inside the cell takes place, then it results in swelling up of the plant cell, and we call this swollen cell as turgid cell. So this is due to the osmotic entry of water inside the cell, and we call it as endosmosis. So in this case, this continuous entry of water inside the cell does not causes the bursting. So why is plant cell placed in hypotonic solution is not bursting like animal cell. This is because there is an extra layer present in plant cell, that is the cell wall. So, the entry of water inside the cell causes some pressure on the plasma membrane, and the equal pressure is exerted by cell wall on the plasma membrane. So, we call this pressure as turgor pressure. So, due to this, the plant cell placed in hypotonic solution remain turgid, so it won't burst. But when the plant cell placed in hypertonic solution, see, in the external solution, solute is more, and solvent is less. So, in such cases, we can see that there is an osmotic exit of water into the external solution, and we call this process as exosmosis, and it results in all the cellular contents comes closer, and plasma membrane also moves away from the cell wall. So, here the plant cell shrink. So, this shrinkage of plant cell when it is placed in hypertonic solution is called as plasmolysis. 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 And when this plant cell is placed in isotonic solution, the osmotic entry of water inside the cell is same as the exit of cell. The osmotic entry of water inside the cell is similar to the osmotic exit of water outside the cell. So, in such case, there is no change in the cell, then this cell is called as flaccid cell. So, it is normal. This is about transport of substance across the plasma membrane, that is passive transport. Under this types of passive transport: diffusion, significance of diffusion, osmosis, types of osmosis, types of solutions, and movement of water in a cell in different types of solutions.